Moi drodzy, uczniowie, studenci, nauczyciele, przyjaciele Uniwersytetu Marii Curie-Skłodowskiej, wszyscy sympatycy nauki i wiedzy. Od dawna na naszym uniwersytecie zastanawiamy się, o czym Maria myśli. Od roku myślimy intensywnie, czym Was zaskoczyć w najbliższym tygodniu. To już od poniedziałku, od 13 marca, zaczyna się kolejny Tydzień Mózgu, na który Was bardzo serdecznie zapraszamy. Mózg to biologia, to budowa, to procesy, ale także od mózgu, od tego, co dzieje się w naszych głowach, zależy to, jacy jesteśmy, jakie są nasze relacje z innymi. Dlatego myślimy o mózgu bardzo szeroko i społeczność Wydziału Biologii i Biotechnologii, Wydziału Psychologii i Pedagogiki oraz Wydziału Filozofii i Socjologii serdecznie zaprasza Was na kolejne bardzo bogate wydarzenia. Chcemy Wam opowiedzieć o mózgu podczas wykładów stacjonarnych i zdalnych, podczas warsztatów, podczas różnych spotkań ze specjalistami z naszego kraju, ale też z zagranicy. Wymyśliliśmy oczywiście dla Was coś nowego. To, co będzie bardzo interesujące, to na pewno warsztaty, podczas których poznacie funkcjonowanie swoich zmysłów w wyjątkowej kapsule do badania zmysłów w Ekotech Kompleks. W imieniu nas wszystkich, a szczególnie w imieniu studentów, wszystkich serdecznie zapraszam. Widzimy się od poniedziałku podczas wydarzeń Tygodnia Mózgu.
Witam wszystkich serdecznie na ostatniej prelekcji dzisiejszego dnia. Naszym gościem jest Gregory Petrazzo, który jest doktorantem w Instytucie Biologii Doświadczalnej imienia Nenckiego w Warszawie. Szczególnie interesuje się badaniami nad starzeniem się i terapiami odmładzającymi, a ostatnio podąża w kierunku zrozumienia mechanizmów łączących mikrobiom jelitowy z mózgiem. Tytuł jego dzisiejszego wystąpienia to Połączenie jelito-mózg w kontekście starzenia się i serdecznie zapraszamy do odsłuchania wykładu.
I would like to welcome everyone to another lecture today. Our guest is Gregory Petrazzo from Nencki Institute of Experimental Biology. Gregory is a PhD student at the Nencki Institute of Experimental Biology in Warsaw. He has a particular interest in aging studies and rejuvenating therapies, and recently moved toward understanding the mechanisms that connect the gut microbiome to the brain. And today, Gregory will present us a lecture on the topic, the gut-brain connection in the context of aging. We encourage you to ask your questions. I will read them to our guest in the end of our meeting. So, Gregory, thank you once again for dedicating your time, and the floor is yours.
Thank you very much for the very kind introduction. Przepraszam, nie mówię bardzo po polsku, albo mówię po angielsku. So good evening, everyone. As you said, I'm a PhD student and I'm actively researching how our gut and our brain communicate and what does it mean for us as we age. So let's start and dive into this exciting world of the gut-brain connection and explore what we can about in terms of aging. Have you ever wondered what the gut has to do with the brain? Well, in our case, it is not the gut itself, but what is inside that matters, the resident microorganism or the microbiota. And let's start by defining what exactly the microbiota is. There is already quite a bit of confusion in the literature, but to be accurate and consistent, we can break things down like this. The microbiota refer to all the microorganisms found in a particular environment. For example, we can talk about the gut microbiota or the skin microbiota and so on. And you may also hear the term microbiome, which specifically refers to the collection of genomes from a microbiota. Each microbiota has a unique composition that can include viruses, fungi, parasites, archaea, and most commonly studied of all, the bacteria. Similarly, each microbiome have a unique genomic signature, and there are specific terms for all the different type of microorganisms that can be used. However, not all these names are commonly used in the scientific community, and in general, people will tend to use microbiota and microbiome to refer specifically to the bacterial component. But if you want to be precise, it's important to use the correct terminology, and I would suggest you to use it like that. So you can already guess that microbiota is complex and a fascinating subject, and we will see how it plays a crucial role in the gut and the brain connection and what happens when we age. Okay, I'm sorry for those of you that are still recovering from the Last of Us final episode, but believe it or not, we are more microbes than humans. And this has been a long-standing debate in the community about the ratio of human cell versus microbe cell in our body. The most recent data that I could find give advantage to the microbiome still, and in fact, only 43% of our cells in our body are of human origin. The rest belongs to microbes. If we look specifically at the microbiome, these numbers are even more striking. We host 2 million different microbial genes compared to our 20,000 human genes. And it is important to note that these numbers are only based on bacterial study. And as you become more an expert in the field by now, you know that there are many other types of microorganisms that can be included in this calculation. So as you can see, there is a ground for the microbiota to play a huge role in the overall makeup of us as a human being. And in this lecture, I will try to explore the impact of our microbiome companions can have on our health, especially in terms of aging. As of today, we know that the microbiota is connected to our brain and can influence our behavior and our mood. But it was not always the case, and you might be curious how this connection was discovered. And to answer this question, we need to take a journey through medical history. In the late 17th century, Anthony von Leeuwenhoek observed that bacteria that at the time he called animalcule in the gut. However, his discovery was mostly ignored for literal centuries. It was only until the early 20th century that scientists began to explore the idea of a gut-brain connection more seriously. In 1907, a Russian scientist named Eli Mechnikov proposed the theory that a gut microbiota could impact our health and even our lifespan. And as you can see, already back then, the press was not very kind with people interested in gut microbiome. But since there have been growing body of evidence exploring the complex relationship between our gut and our brain. And it was clear actually for many physicians throughout time that there was some level of connection between our emotion and our internal organs. For example, when you are with your significant others and you start to feel butterflies in your chest, or when you have to give a very long lecture at Białystok University for Brain Week and you feel a ball of stress in your gut. Well, some understanding of this process come from a current of thought that spanned throughout the whole 18th and 19th century. Robert White, in 1765, observed the presence of a huge amount of nerves in the intestine, and he believed that this system was connecting all the organs together by what he called a nervous energy. Paul-Joseph Barthès and his students Jean-Charles Grimaud developed the idea of a vital principle that distinguished later into a principle of an animal motor force responsible for sensory function that are promoted by the brain, and a vital motor force that was connected with essential body activities to survive, such as nutrition and respiration, the things that your body does without you consciously making it happen. And this current of thought led Marie-François Xavier to his own current of thought that was vitalism. And in vitalism, Marie-François Xavier really stressed the importance and the connection between the gut and the brain. And Robert White was also very influential for one other person later in time, that was John Abernethy, who published several books linking mental disorders to nervous disorders of the stomach. He pointed out that disturbance in the stomach that were the cause of sleep and mood abnormalities. And his work was so influential that already at that time, the stomach was regarded as a great abdominal brain by physicians. But it became more than just linking mental disorders to the stomach. And I will give you a bit of trivia. In the 19th centuries, England population was very poor, and many people from the working class relied on the tea and bread consumption to sustain themselves. It was especially true for women at the time, and this became at some point a public and political issue, to the point that the Dean of Bangor, Henry Thomas Edwards, in 1883, claimed that the tea consumption of the working class led to nervousness, hysteria, and discontent, with poor control of their negative emotion. The tea was even marked as a dangerous revolutionary force that seemed to be swelling into a flood of radicalism and lead to lamentable immorality. It was clear for them that drinking an excitatory drink might deregulate the nerve in the gut and influence the mood. Another example is during the industrialization period. Gut diseases were a major national issue and were described for the upper class as a result of overthinking and a sedentary lifestyle, while for the working class it was seen as a result of poor diet and hygiene and alcohol consumption. No matter the political agenda, it was clear that there was some connection between the state of the gut and people behavior, but still not so much interest on the microbiota, until physicians like Charles Bouchard in 1887 started to link cause of auto-intoxication coming from toxin produced during the digestion and not eliminated fast enough with the presence in the gut of microorganisms. If you remember Anthony von Leeuwenhoek from just a few slides ago, we are now around 300 years later, and the idea of microbiota start to bloom with the direct observation in the gastrointestinal tract from many physicians in different countries. One that you might be more familiar with is this person, even if you are not aware of it, because Theodor Escherich was the person who observed and described a commensal bacteria that will be named after him, Escherichia coli, that we all very well know. From there, scientists have identified many different types of bacteria in the gut and elsewhere, and have discovered dozens examples of how these microbes can impact our health, and it has sparked a lot of excitement and momentum in the field of gut-brain aging and microbiota research. And actually, the term human microbiota was only popularized during the 21st century and truly conceptualized with the creation of the Human Microbiota Project in 2007. It is a significant milestone that aimed to identify the composition of the human microbiome and the role of each microorganism in it. And as you see, it took a lot of time, a lot of effort, and collaboration between different countries to establish the link between distant organs, identify various players involved, and understand how they interact. Of course, I only touch on the tip of the iceberg here regarding the history of the field, but if you are interested in learning more, there is a wealth of information available online that you can provide you with deeper understanding. Let's switch gear a bit now and delve into how we can actually analyze the microbiome and study the gut-brain axis. In the past, not so long ago, but still in the past, from our point of view, identifying bacteria in an environment involved collecting a sample and then putting it into a culture in a petri dish to isolate a single type of bacteria, and that allow for later, when you have it isolated, to identify it with biochemical techniques to really say, okay, I have found one bacteria. However, now we have also new technology that allows for more sophisticated technique than just biochemical and enzymatic approach, like the MALDI-TOF, that is a mass spectrophotometer that uses a laser to ionize a sample, and it will record a specific spectrum that can identify a specific bacteria. With this method, they are useful, but they can be very time-consuming. Culturing bacteria take a couple of days. Not all the bacteria can grow in every medium. And there is an initial significant cost if you want to buy a Malditov. So instead of relying on these traditional cultures, we now have access to genetic approach that enable us to analyze the DNA and RNA of our microbiota. By looking at the DNA, we can determine the composition of the microbiota by clustering all the bacteria based on a highly conserved DNA sequence that is shared among one species. This can be incredibly useful in identifying bacterial species that may be associated with certain disease and phenotype, but mostly also for bacteria that are really hard to put into culture. We can also analyze the RNA, and it helps us to understand how the microbiota can sense and react to change in its environment. For example, a change in the diet or the use of an antibiotic can trigger a shift in the bacterial community, and studying the RNA gives us an insight into how they respond to this change, and probably also on how it can affect the body. Of course, genetic approach can also be used in combination with protein and metabolic-based approach to provide a deeper understanding of how the microbiota interact with the host. This approach gives us a clearer picture of how the body senses the bacteria and how the immune system should react to it. But also their activity may have an influence on distant parts of the body, such as the brain. And by harnessing all these innovative methods, we can gain a better understanding of the intricate relationship between the gut microbiota and its host, and how this dynamic interaction can impact the health and well-being of the people. So let's take a practical example. Suppose we want to investigate the effect of a low-carb versus high-fat diet on the microbiota of a rodent model. To do this, we can feed our animal with their specific diet for a period of time. We can collect the sample for the microbiota studies, and this sample can be from different sources, from different parts of the intestine or feces or whatsoever. After obtaining the sample, we can extract everything from it: DNA, RNA, and proteins. If we want to study the DNA, we can amplify a specific region. Usually we use the 16S ribosomal RNA gene because it is very well conserved among species. So it's a very good target to identify the different species. Once we have it isolated and purified and amplified, we can send it for sequencing. This can be done at the university core facilities. Some research institutes have some private company does it also. And once it has been sequenced, you receive a lot of information and raw data that can be put into bioinformatical tools, and this will allow to do a lot of things. We can determine the number of different species in different groups. That would be the alpha diversity. We can also determine how many species are different between several groups. That would be the beta diversity. We can also determine the proportion of the different genera, phylum, and species present in one sample. That's what we call the taxonomy. And we can also associate certain bacteria, their presence, with other parameter in the body. Like in our case, it could be weight gain, fat accumulation, and so on and so on. And to investigate further, in that case, we can also analyze the RNA and the proteins, and that will give us a whole idea of how change in the diet impact the whole body. So if you want to specifically address questions regarding the gut-brain axis using our rats, to begin with, it is necessary to characterize the cognitive phenotype of the animals. This involves feeding them with specific diet and then conducting behavioral tests using various equipment. And this equipment allows us to actually address different type of cognitive function. We can look at anxiety, pain, social behavior, motor function, learning, memory, things like that. And we can do that by using different tools. We have a different type of maze. We have a rotarod, which is a forced wheel walk. We have social interaction cages. We have LiDAR pool cages that allow us to look for anxiety. And this helps us to better understand the complex interactions that are happening in the brain. Now, in terms of microbiota, to investigate the impact of it on the behavior, we have several ways to do that. We can entirely remove the microbiota from our models. There is two ways of doing that. Either we give a broad spectrum of antibiotic to our animal and we keep them on it throughout the whole experiment, or we use animals that we call germ-free animals, which are specifically bred without microbiota. Although the germ-free animals are quite expensive, they require special housing, they are very viable tool to study the impact of the microbiome on behavior. Once we have given our regimen to our animals, we can again test them for the behavior and compare the result with animals that have their microbiota intact. And that allows us to determine whether a change in diet that can have an impact on, let's say, for like capacity to run or capacity to solve a problem or memory, if that change on diet and that improvement in cognitive function or exercise is dependent or independent of the microbiota. Another way of doing studies on the gut-brain axis is to set experiments that involve transplanting the microbiota from one animal to another. First, you can give your specific diet to your animal, and then you collect the microbiome and you transplant that microbiome to receiver animals. And these animals, let's say, are just fed on normal diet. So you can just see really the impact of the transplanted microbiome. And after that transplantation, you can again characterize your animals for enduring behavioral testing. And this paradigm is useful to determine if the microbiota can independently cause changes in the behavior and in the mood. For example, in our high-fat diet case, maybe the bacteria in the microbiota could sense the change in the diet and produce metabolites that will create more inflammation in the host, and ultimately that could lead to an increased anxiety. And the main advantages of performing this type of experiment is that it allows us to isolate the specific contribution of the microbiota on the host. If instead of a diet, we were giving the rat drugs that have multiple organs as a target, including the microbiota, performing this type of transplantation could help distinguish what effect comes from the drug and what comes from the metabolism of the microbiota. So now let's talk a bit about aging. I have teased it a bit too much from now, which is something that I guess everyone now will experience as we grow older. There are actually two types of aging that we should distinguish: developmental aging and senescence aging. And the former refers to change that happen during the course of our normal growth and development, while the latter is characterized by a progressive decline of biological function and decreased ability to adapt to metabolic stress. Now, some of you may be wondering, why should we study aging since it is a natural process and not a disease? Well, the truth is that it never has been, it has been like, again, a huge debate in the scientific community, because aging has been considered a disease for a long time. Then it was considered as a natural process and then not a disease, which made the grant application for many research labs very difficult. And now it's again classified as a disease. It's almost like the Pluto of biology, constantly shifting in its categorization. But despite these debates, the study in aging is incredibly important. As we age, our bodies undergo a number of changes that can have significant impacts on our health and well-being. And by understanding these changes, we can work to develop interventional therapies that can help us maintain our health and independence as we grow older. In particular, understanding how the microbiota can affect aging is an area of active research, as changes in the gut microbiota have been linked to a variety of age-related diseases, such as Alzheimer and Parkinson and dementia. Nevertheless, I will try to continue to convince you that it is important to address this field of research. For one thing, is the share of elderly people in the world is expected to rise dramatically in the next few decades. And it is absolutely evident that aging is the main variable for age-related diseases. And you can think again as diabetes, cancer, metabolic syndrome, etc., etc. For the brain, it is associated with dementia, cancer, Alzheimer, Parkinson, and cognitive decline in general. And it is almost a philosophical argument whether it is best to study and treat, let's say, dementia alone, for example, or try to tackle what is at the root of all this disorder, which is aging. And because if you delay aging, you could also delay the subsequent appearance of all these conditions. And to do so, it is necessary to understand the key biological mechanisms of aging. And today we have well-defined hallmarks of aging, the same way that there is hallmark of cancers. And according to this aging hallmarks, it's like a combination of internal damage, such as genomic instabilities, telomere attrition, epigenetic alteration, loss of proteostasis, that in response to this damage, the cells start to have deregulated nutrient sensing, mitochondrial dysfunction. They will turn senescence, which is a state where the cells usually don't divide, but produce a lot of harmful signaling molecules. And all this leads to stem cell exhaustion and intercellular communication pathway, an altered intercellular communication pathway. When such event starts to appear, the tissue will progressively be impaired until it cannot function correctly, and you will see the appearance of various symptoms. So these hallmarks were published, I think, in 2013, but since then there is new hallmarks that were added, and that camera is going a bit wide. Okay. And so these are mostly internal process, but now we have like also more external process that come into play, and that include inflammation and microbiome disturbance. And I spoke of inflammation as a marker of aging because it is well known and described now that while we age, we see an increase in inflammation marker in the body. And this process has been termed as inflammaging. This notion is, in my opinion, is very central to the aging process, and it's in link with the microbiota. For example, here you can see in this graph that they compare the life expectancy of wild-type mice in solid line with germ-free mice in dotted line, and you can see that the germ-free mice tend to live longer than the wild-type mice. And that has been summarized here, that while we age, we see an onset of dysbiosis. The composition of our microbiota changes. More harmful byproducts are metabolized, and the intestine barrier becomes more leaky. And it creates a systemic inflammation when gut-derived metabolites pass through. This increase in inflammation may lead to senescence in the body and therefore promote the appearance of disease. And the literature is now very rich with data from adults, from elderly, and even centenarians when it comes to microbiota and aging. What this paper shows is that there is a strong relationship between the gut homeostasis, the level of inflammation, and the lifespan. There is a loss of diversity during aging that starts to appear. Some species become less abundant with advanced aging, and some clusters remain very well present, like with Actinobacter here in the centenarian group. And it could be linked with a particular lifestyle that may explain in part why these people live that long. But if you think about our animal model, They are kept in a strictly regulated environment with controlled light, controlled humidity, access regularly to food. So their microbiota should be very stable over time. And in reality, we see exactly the same onset of senescent microbiota in these animals too, which means there is some loss of homeostasis in the body that may impact the living environment of the bacteria in our gut. And going back to the lab and using the experiment paradigm that I explained a bit earlier, in this research on the left, the research team took the microbiome of young mice and transplanted into aged mice. And it was able to reduce the systemic level of inflammation. But what happened in the brain? The mice that had the young microbiota transplanted performed better in behavioral tests. They were able to perform better in maze, where we look at the spatial cognitive abilities, and they show it was associated with a decrease in neuronal inflammation, shown by a decrease in microglia activation. And they also show that they restore hippocampal metabolites, which may explain why the animal performed better in the behavioral test. Another example here, where they show the same kind of result from a young microbiota transplanted into aged mice. There is less systemic and neuroinflammation, but moreover, they did the opposite. They transplanted aged microbiota into young animals, and they show that these young animals now show increased systemic and neuronal inflammation. In my lab, we are interested in the senescence aspect of aging. I started my PhD working on senolytic drugs, which are drugs that specifically target the senescent cells to remove them from a tissue. What we did was very similar to what you just see. We treated aged rats with senolytic drugs, and we tested them for behaviors in inflammation. We observed that when you give the senolytic treatment to the aged rat, they have better memory and learning capacity, and they have reduced inflammation. And all in the brain, what we see was a change in the synaptic plasticity. There was a remodeling of the neuronal network that may explain what the improvement in the behavioral apparatus. So naturally, I decided to investigate what are the changes associated in the microbiome. And there is some that are consistent with the literature. So currently, I'm trying to investigate what are the communication routes from the microbiota to the brain. And what is important to understand concerning the communication routes between the gut and the brain, it is a bidirectional communication. I show you that changes in the microbiota composition and metabolism can affect the behavior. We discuss also how the changes in the body homeostasis can affect the microbiota. Therefore, there is some mediator that will transmit a signal from one or the other. One of the primary mechanisms is the vagus nerve. It is a nerve that runs from the brainstem down to the gut. This nerve transmits signal bidirectionally between the gut and the brain, and it plays a critical role regulating various physiological functions, including digestion, nutrient absorption, and gut mobility. That will dramatically impact the environment of the bacteria and then their growth and their metabolism in general. Another important mechanism in the gut-brain axis is the production of neurotransmitter and other signaling molecule by the gut microbiota. These molecules can interact with nerve cells in the gut and in the brain and influence the neuronal activity and altering the behavior and the mood. For example, the gut microbiota can produce neurotransmitters such as serotonin and dopamine, which are known to regulate mood and behavior. The gut also produces a variety of hormones that influence brain function, including ghrelin, leptin, and insulin. These hormones play an important role in regulating appetite, metabolism, and energy balance, and they can also, as you have guessed, influence cognitive function and mood. As we discussed earlier, also, the immune system plays a critical role in the gut-brain axis. As the gut is the home of a large number of immune cells, these cells produce cytokines and other signaling molecules that can influence neuronal activity and alter the behavior and mood. For example, inflammation in the gut has been linked to depression and anxiety. One very interesting gut-derived metabolite that is actively involved in the gut-organ intercommunication is the short-chain fatty acids. These are groups of metabolites that include acetate, propionate, and butyrate. They are produced by microbial fermentation of dietary fiber in the gut. They have been shown to regulate and release various neurotransmitters, including serotonin and dopamine, which are involved in mood and regulation and cognition, as I said. And they are also can act as signaling molecule by themselves to modulate immune function and inflammation, which can again affect the brain if it goes through the blood-brain barrier. Furthermore, studies have found that these short-chain fatty acids can influence the integrity of the gut and the gut barrier and of the blood-brain barrier, which can then promote or decrease the passage of substances from the gut to the bloodstream and to the brain. And these can be particularly relevant in the context of neuroinflammation and neurodegeneration, where the gut barrier dysfunction may contribute to the development and progression of brain disorders. They have also been shown to promote the growth and survival of neurons, which can help maintain cognitive function and memory. They have been shown to decrease neuroinflammation, stabilize brain homeostasis, and improve the blood-brain barrier integrity. So overall, these short-chain fatty acids play a crucial role in the gut-brain axis by regulating neurotransmitter release, immune function, hormone secretion, and physiological barrier integrity. And further research is absolutely needed to fully elucidate the mechanism underlying this effect, especially in different types of pathologies. So I hope I convinced you so far that the microbiota can be both a marker of something happening in the body, but also as a therapeutic target to influence a process in the body. And it is not a very new concept. If you remember Elie Metchnikov from the beginning of this lecture, he observed that some bacteria prevented milk from rotting. He proposed that a similar process of putrefaction was happening inside the human gut and could be responsible for premature aging. He then hypothesized that some bacteria may be able to reverse aging, and that was more than 100 years from now. He went through with his idea and developed what would be the first precursor of modern probiotic in Paris in 1905 from lactobacillus cultures to fight aging. And I guess that people that would be around my generation are very well aware of what are probiotics and prebiotics, but in fact they are a form of gut-targeted therapies, with probiotics being live organism culture from fermented food or in supplement and prebiotics being food that will help increase the proportion of certain microbes. And because it is a rapid growing industry, we see now more and more study and commercialization of what they call psychobiotic, which by targeting microbiota are supposed to have direct effect on the brain and therefore on the mood and behaviors. At the moment, there is a lot of effort that is made into precisely describing the microbial communities from different lifestyles, addressing how it impacts the gut-brain axis, and trying to formulate the best-looking compounds to promote healthy aging. I am sorry if it was a bit shorter than you expected. I hope you still have enjoyed this lecture. I will be happy to discuss with you if you have any question, and if you want to know more about it, please shoot out.
Wow, thank you so much for such an interesting lecture. It was really amazing.
Wow, I'm really happy you like it.
We have several questions from our audience. So the first one is it true that everyone has a unique composition of microbiota and it depends on our diet in the childhood?
Okay, so you can see actually the microbiome as a fingerprint. It is quite unique, but it's quite unique to the point that there will be similarities, especially within families or within people that live close together. If you live with your parents, you are more likely to have a microbiota that is more similar to them than if you talk with your friend that is living in the next city. For sure, the early onset of microbiota graft in the body can stay very long in the body. So it change over time. That's one thing for sure. Your microbiota is evolving as you age, and it is very likely that what happened at the very beginning can have long-lasting effect and can stay longer.
Okay, thank you for your answer. So did I get it right? It also depends from genetics and from our lifestyle, right? So both.
Yeah, alors, not so sure about genetic exactly. I would more say that maybe epigenetic will have more to play than genetic per se, because it's all a matter of homeostasis in the body that can change the fingerprint of your microbiota. Lifestyle will have the more dramatic effect because what you eat will directly goes there and will directly be used by your microbiome. And yeah.
Okay, thank you. Our next question is, how is it possible that our immune system doesn't detect this microorganism in our gut as something wrong?
Okay, so we have evolved with this microorganism for a very long time, and they have adapted very well to their environment in our gut. They are producing a lot of mucus that help them to grow and stay there. And you have that physiological barrier in the intestine that separates them from your body. So in normal situation, everyone is of one side of the fence or the other, but the bacteria made their own environment to grow and stay there hidden and in good term with our immune system. It's only when you start to have disruption of that barrier and that some components start to pass through the barrier that the immune system will not be happy and will start to target them. But otherwise, they adapt very well there.
Okay, thank you. Our next question is, so what are the top products to eat to keep our microbiota healthy and live longer life?
That's the $1 billion question. And if I had the answer, I'll be running a very successful company by now. But, you know, there is always that debate, processed food versus natural food. I would suggest to, without medical advice, but I would suggest people to go with fermented food, a bit like what we can see from Asian culture, from Japan, Korea, and so on. They eat a lot of fermented culture, and in this culture you can find either probiotics or it is a source of prebiotics that tend to increase the kind of good guys in the gut. Anything that's fermented. And even in Poland, I know you have a lot of fermented food from cabbage, and it's actually very good.
Okay, fine. So no sweets?
I'm not a big fan also personally, but the studies with a lot of sugar in the diet or a lot of fat in the diet, they tend to increase the bad guys.
Okay.
I'm sorry.
I'll survive with that information somehow, but okay, our next question is improving our microbiota can help us in memory loss, especially after COVID infection?
I haven't looked at that, but I know there is a lot of studies right now on the COVID and the microbiome, and for sure on the brain fog from the COVID. I would assume that yes, because most of the studies, when you try to look at something plus the microbiota, there is an effect. So my guess, and we'll have to check, but I'm pretty sure it will be like that. There would be some microbiota targeted therapy that would help to alleviate some of the burden of the COVID brain fog.
That's interesting. Thank you. And I think our last question for today is, Are there any specific protein modifications connected with microbiome which accelerate aging?
Oh, that's a very good question. I haven't looked at that in particular, but my guess is as good as any, but I would probably think there would be something like that because, you know, gut is producing a lot of metabolites that if they interact with the cells, they will deregulate the protein homeostasis and probably also the protein post-transcriptional modification. That is something to see, but that's the problem that you will have to address is okay with which cells where, and you will have to find then which bacteria and what compound from this bacteria does the job. That would be a very long research project but very interesting.
Okay, thank you. But I assume that advantages of healthy microbiome definitely are greater than disadvantages of a possibility that something can cause the acceleration of aging, right?
For sure. I mean, it's really the chicken versus the egg question. So what starts first and what drives the other first? So is the microbiome driving the aging because it becomes an essence, or because we age, we start to have a bad microbiome, and because it's bad, it's just accelerated everything in a bad, in a vicious circle? That my suggestion is try to eat the best food for your gut that will help you age the best, and try to be centenarians.
Okay, we'll do our best, I suppose.
Sorry, no sweets. But wine can be okay because it has a lot of polyphenols, and polyphenols are good for the gut, and it's good for aging.
Okay, that is a good news.
At least.
Okay, then thank you very much, Gregory, for your interest and I assume practical lecture and for your willingness to join our event. It was a great pleasure.
Thank you for having me. Thank you for all the questions. If you want to reach out to me, I guess there you can go through the organizer. I'll be happy to reach with everyone.
Okay, thank you so much. And thanks everyone for your attendance today. And we would like to invite you to join our tomorrow's workshops and lectures. Goodbye. Have a nice day, everyone.